Swayam Prabha. Digital India. Educated India.
The topic for discussion now is carrier proteins on the cell membrane. The context is this: we considered cell membrane proteins in 6 groups based on their function. The mnemonic is here, TREXA, standing for transporters, receptors, enzymes, G proteins, structural proteins, and adhesion proteins. We then focused on transporters on the cell membrane. When we consider transport processes per se across the cell membrane, transport can occur through simple diffusion via the lipid film, and that can be true only for lipid-soluble substances. Whereas polar substances or hydrophilic substances require the assistance of membrane proteins called transporters to take them past the membrane without seeing the lipid. And a third transport process is exocytosis or endocytosis. We are right now concerned with transport proteins on the cell membrane. The transporters on the cell membrane can be broadly classified into ion channels and carrier proteins. Dr. Vinayuman has dealt with ion channels in some detail, and now we are moving over to carrier proteins. The carrier-mediated transport processes in the cell membrane can be classified in 4 ways: facilitated diffusion, secondary active transporters, primary active transporters. And this 4th group is not usually discussed in undergraduate textbooks. Nevertheless, this is a very important class, ABC transporters, the ABC here standing for ATP binding cassette. This group is very important in cancer therapeutics, and we will consider them in some detail here. Dr. Vinayuman had grouped transport processes slightly differently. This is the classification he had used, and these come under the carrier-mediated transport systems. To this group, I am adding on ABC transporters. So, this is the group that we are concerned with now. Now, in this classification, he had said it well that transport through ion channels is simple diffusion. There are some school textbooks which refer to diffusion through ion channels also as facilitated diffusion. That is not entirely correct, because the term facilitated diffusion is technically reserved for carrier-mediated transport processes. What is the difference between simple diffusion versus facilitated diffusion? The difference is in the kinetics of transport. Before we discuss the difference between these two systems, let us revisit diffusion through ion channels and understand that better. Here's a cell, and let us say that is a potassium channel on the cell. Potassium concentrations in and out are like this: 150 inside and 5 outside. This is the resting state in the cell, and there are constitutively open potassium channels. Does this mean that there is continued diffusion of potassium from in to out in a resting cell? If that is the question, the answer is not necessarily so, because remember, the resting cell has a potential difference across its membrane, what's called the resting membrane potential, and that we saw for a neuron would be about minus 80 millivolts. So, while there is a concentration gradient directed outward for potassium exit, the electrical gradient, being negative on the inside, will not permit potassium efflux. And at the resting membrane potential, the potential that has developed balances the concentration gradient exactly, and even though the channel is open, there is no net efflux of potassium. In fact, we will see later that this potential difference itself is nothing but the diffusion potential or the Nernst potential for potassium given these concentrations. So, in a resting cell, the electrochemical gradient for potassium movement across its ion channels is nil, and therefore there is no net efflux or influx of potassium. So, what you need to understand is that for diffusion through ion channels, what matters is the electrochemical gradient and not just the chemical gradient. The electrochemical gradient in this scenario is 0, and therefore there is no net diffusion of potassium across the membrane. Now, if the cell membrane voltage became more positive, let us say from minus 80 it moved to minus 40 millivolts. Now the inside being more positive than the equilibrium status, potassium will move out of the cell. Outward currents are designated as positive currents by convention. On the other hand, if the inside of the cell became more negative, one way of doing this is experimentally by clamping the voltage with a patch clamp amplifier. If the membrane voltage became more negative than this equilibrium value, then there would actually be net influx of potassium. This should not be construed as active transport from a low concentration to a high concentration, because what matters is the electrochemical gradient, the internal negativity being much more than the energy of the concentration gradient. There is the energy gradient or the electrochemical gradient for potassium diffusion is now directed inward, and there would be an invert current. And invert currents by convention are designated as negative. If we were to depict this in what is called an IV curve, where the membrane voltage is taken along the x-axis and the current on the y-axis, engineers would want to call this a VI curve because voltage is on the x-axis. However, in electrophysiological terms, it is referred to as an IV curve. This point here represents the fact that there is no current when the membrane is at the resting voltage, that is at minus 80 millivolts, no potassium current through its channels. And if the membrane becomes more positive, if it moves to about minus 40, there is an outward current, and as I said, that is designated as a positive current. And if the membrane becomes more negative, there is an invert current through potassium channels, and that is also diffusion, simple diffusion. Now, having considered the profile of flux across the cell membrane through ion channels, we will now see what is the difference in kinetics between simple diffusion and facilitated diffusion. Let us look at the kinetics of diffusion through an ion channel. Let's take a sodium channel now. The external concentration of sodium is 140 millimoles per liter, and within the cell it is only about 14 millimoles per liter. And even at rest, because the inside is negative, there is not only a concentration gradient for sodium in flux, but also an electrical gradient. There is a net electrochemical gradient for sodium in flux if the sodium channel is open. And if we can estimate flux or the amount transported per unit time across the membrane, flux can be measured in various ways. We can either put a radioactive tracer molecule here and then look at how much of radioactivity has entered the cells. Or we could look at currents, ion currents in a patch clamp experiment. So, if we had an estimate of the flux across the membrane, we would see that as the concentration of sodium on this site increases, the flux would also increase. If we plotted the EC of sodium concentration on the x-axis and sodium flux on the y-axis, we would see that as we increase sodium concentration on this side, flux will increase linearly. So, this kind of transport where the flux increases indefinitely or keeps increasing linearly as you increase concentration gradient is called simple diffusion. In contrast, let us see how flux is related to concentration gradients in the case of facilitated diffusion. Not just facilitated diffusion, any carrier-mediated transport process. Let us take glucose as the example. The glucose transporter, or Glute as it is called, mediates glucose diffusion across the cell membrane. Glucose would bind to one side. There is no through and through conduit for glucose to go across within this protein as it occurs in an ion channel. After glucose binds to the external face of the protein undergoes conformational changes, literally squeezing glucose through the membrane to the inside, a process which has been elegantly referred to as even molecular peristalsis. So, the protein has to do work. It has to undergo conformational changes to bring glucose from one side to the other side. And if we were to plot flux against concentration gradients, we would see that initially, as we increased glucose concentration on this side, flux would naturally increase because more and more glucose is available and that would bind to as many transporters as there are available. But after a certain level, even if you increase the glucose concentration on this side, flux will not increase any further and will level out, because now all the transporters are occupied by glucose and they have to undergo conformational changes, and that becomes the rate limiting step now. There is a finite speed with which these conformational changes can occur. So, the protein has to move glucose inward and then flip back to its position to take up another glucose. This is what leads to saturation of the transport process, what is referred to as saturation kinetics, much like the enzyme kinetics. Or this transporter does what an enzyme does to a chemical reaction. It has to bind the substrate and facilitate the reaction. Based on this fact, we classify diffusion through ion channels as simple diffusion, where there is a linear relationship between flux and concentration gradient, and facilitated diffusion through carrier-mediated transport systems as something which demonstrates saturation kinetics. We have just learnt that carrier-mediated transport systems exhibit saturation kinetics. We will now look at general characteristics of these three types of carrier-mediated transport processes. In facilitated diffusion, the substance moves along its concentration gradient. These are generally unipotes, which means they transport only one substance along the concentration gradient. The glucose transporters is the best example for this class of proteins. The other two important proteins are amino acid transporters and urea transporters, which also demonstrate facilitated diffusion. In primary active transport, the substance to be transported moves against its concentration gradient. These are either unipotes, where there is only one substance that is pushed against a gradient, or they could be antipodes. They could transport two substances in opposite directions, both of them against their concentration gradients. If the substance has to move against a concentration gradient, then the transport process requires extra energy, biological energy, and that comes from cleaving adenosine triphosphate. The transport protein itself is an ATPase enzyme. It has a binding site for ATP. It would cleave ATP energize itself and undergo the conformational change to push the ion that is to be transported against its concentration gradient. So, all primary active transporters transport only ions, not non-ionic substances. So, these could be either unipodes transporting just one substance or antipodes. In secondary active transport, one substance, not necessarily an ion, would move against its concentration gradient, but the energy for uphill movement of this substance does not come from cleaving ATP directly. Secondary active transporters are not ATPase enzymes like primary active transporters. The energy for uphill transport comes from downhill movement of another substance along its concentration gradient. This is usually an ion. In fact, it's almost always sodium that moves downhill via its gradient into the cell, and downhill movement of sodium would be an exergonic process releasing energy, and that energy is harnessed for uphill transport of another substance. So, secondary active transporters can never be unipodes, they always have to transport two substances. So, if both the substances move in the same direction out to in, we call them sympodes or co-transporters, and if they were to move in opposite directions, we refer to them as antipodes or counter-transporters or exchangers. The preferred term would be exchangers if it is secondary active transport. So, you must be careful not to use the term exchangers for primary active transport. For example, the sodium potassium pump, you can't call it a sodium potassium exchanger. It is generally not referred to as an exchanger. The primary active transporters are better called as ion pumps or ion translocating ATPases. And please don't call any of these channels. Students have a general tendency to use the term channel, pump a little indiscriminately. So, we have to be very specific in usage of these terms. When we refer to an exchanger, we are referring to a secondary active transport process, and when we refer to a pump, we are referring to a primary active transport process, and when we say channels, we are referring to simple diffusion. Now, when it comes to learning about a carrier protein, we can ask a set of questions. Remember, Dr. Vinay Uman gave you a set of questions that you could ask for about an ion channel to understand more about it. Similarly, if we think about a carrier protein, we could ask these various questions. What is the type of transport? Is it facilitated diffusion or primary active transport? What is the substance that is transported? What is the stoichiometry of transport? What do we mean by that? If it is a sodium potassium pump, we know it pushes out 3 sodium ions and takes in 2 potassium ions for every turn of the transporter. So, stoichiometry refers to the number of molecules of the substance that are transported per cycle of conformational change of the transporter. Then you could ask, what is the direction of transport of these substances? Does it go into the cell or out of the cell? Where is it located? If it is located in epithelial tissue, is it located on the luminal site or the basolateral site? Is it electrogenic? What is the clinical significance of this transporter? Are there disease processes which happen due to faulty transport? Are there therapeutic drugs which modulate these transporters? Are there side effects of drugs because they modulate some transporters? These are all questions you should keep in mind when you're learning about a transport system. Now, some of you may wonder, why go into these ecstasies about membrane transporters? Let us see some numbers to understand the relative abundance of membrane transport proteins among all proteins in the body. Now, proteins are coded for by genes. Genes are DNA sequences coding for proteins, and genes per se form only less than 2% of the entire genome. The entire genome is made up of 3.3 billion base pairs, and only less than 2% of them, that is about 30 to 40 million base pairs, actually form genes. That is the coding region of the DNA. The rest of it is non-coding region made up of introns, regulatory sequences, RNA coding sequences, etc., etc. So the protein coding sequences, which are also referred to as the exome, is less than 2% of all the DNA. So somebody has taken up these 19,000 genes coding for proteins and has worked out a pie chart to show the relative abundance of each class of proteins. That's the reference. And we are now looking at transporters on the membrane. So this is where it is. So the transporters form about 6.4% of the proteins. There are about 1,000 genes which code for transporters amongst this whole set of 19,000 genes. And yes, we have receptors here. That is an equal number. Now, I would like to draw your attention to this term here, carrier proteins. It's used in a different context, and I thought I would use this opportunity to highlight the other context in which the term carrier proteins is used. Here they are referring to plasma proteins which bind to other substances and carry them in blood. For example, albumin binds to bilirubin, and that's how bilirubin is transported in blood. So the term carrier proteins is used in that context here. What we are referring to are membrane transporters. We called them carrier proteins as well, or carrier mediated transport proteins. So let's look at transporters. Here they say 6.4% of all proteins, and there are other estimates which say that they form about 10% of all proteins. While membrane transporters and membrane receptors form only about 15% of all the classes of proteins in the body, you would be surprised to know that 50 to 60% of all therapeutic drugs that are available today actually act on membrane transporters and receptors. And therefore, it is important to invest some time in learning about the details of membrane transporters and membrane receptors. So, these are the membrane transporters which form, let us say, 10% of all proteins in your body. Now, functionally, or rather in terms of gene names, they can be grouped. These two can be grouped as solute carriers. Their genes are named as SLC1, SLC2, etc., etc. So there are four groups now: ion channels, solute carriers, which includes facilitated diffusion and secondary active transport. Then we have the primary active transporters and the ABC transporters. So here's a pie chart from this reference of the relative abundance of these four transporter types. I've modified this slightly. So ion channels form about 40% of the transporters. They come from about 300 genes. Solute carriers, which includes facilitated diffusion and secondary active transport, make up about half the number of membrane transporters. That's the percentage of primary active transporters and then the ABC transporters. Just because they are less abundant, it does not mean that they are less important. In fact, today there's as much research going on about ABC transporters as about ion channels. We will now move on to a discussion on facilitated diffusion mechanisms. Glucose transporter is the best example for facilitated diffusion. We already saw that glucose binds to one face of the transporter, and the transporter has to undergo conformational changes to release glucose on the other side. There are also amino acid transporters and urea transporters. we will discuss a little more about glucose transporters. There are about 14 subtypes of the glucose transporters, but the first 4 have been known for quite some time, and the others have been recently identified after the Human Genome Project. We'll look at glucose transporters 1, 2, 3 and 4. I have shown the blood compartment here because glucose moves from blood into extracellular fluid and then into cells. The concentration of glucose in blood is about 5 millimoles per liter. And glucose transporter 1 is located on the cell membrane, which would take up glucose from the ECF and move it into the cell along its concentration gradient. Glute 1, in my understanding, is an ubiquitous transporter present on almost all cells of the body. It is a low volume glucose transporter, ensuring a baseline constitutive uptake of glucose for the energy needs of the cell. There is always a gradient for glucose to move into the cell because once glucose enters the prototype cell, it would get metabolized. It would either undergo glycolysis or be converted to some other substance. So, the cells are always sinks for glucose, and there is always a concentration gradient for glucose to move from blood into the cells. Glute 3, as opposed to glute 1, is a high volume glucose transporter and is located in brain tissues, because brain usually has priority over other tissues, and this high volume transporter will ensure that the brain gets its share of glucose even while blood glucose is dropping. Glute 4 is found on striated muscle, skeletal muscle and the heart, adipose tissue and brain. Glute 4 is the insulin dependent transporter. We will come to that a little while later. Glute 2, on the other hand, is found in liver cells, pancreatic beta cells, epithelial cells of the intestine and epithelial cells lining the proximal convoluted tubule of the nephron. Why did I choose to show glute 2 on this other side? Because my understanding is that while glute 1, 3 and 4 are unidirectional glucose transporters, taking glucose from the blood into the cell, glute 2 is a bidirectional transporter, because there are situations when the cell has to release glucose to the outside. Let's take the example of a liver cell. When glucose is abundant in blood, the liver cell will take up glucose through glute 2 and convert it to glycogen, a storage form of glucose. And when there is less glucose in the blood, this cell will release glucose into blood. Therefore, there is a need for bidirectional transport. The pancreatic beta cell 2. The pancreatic beta cell is the glucose sensor, which has the job of sensing the amount of glucose in blood and therefore modulate insulin release. Therefore, its levels should equilibrate with external levels if it has to be a glucose sensor. And therefore, it is important to have this bidirectional transporter on the pancreatic beta cell 2. Now, let us look at the location of glute 2 in the intestinal or the renal tubular epithelium. This is the PCT. Glute 2 is found not on the luminal border of this epithelial cell, but on the basolateral border. What's the purpose? Because in the renal epithelium, in the primary convoluted tubule, glucose has to be absorbed from the lumen into the cell, and that transport is through a different transporter. We'll come to that later. And from the cell, glucose diffuses out into the extracellular fluid through glute 2. Now, this transport of glucose from the lumen into the cell, because it goes on constantly as long as there is glucose in the lumen, the cellular concentration of glucose is high and Glute 2 will ensure facilitated diffusion of the glucose from the renal epithelial cell into the ECF and then into blood. So, this is involved in reabsorption of blood glucose, of filtered glucose, or the glucose in the diet in the case of the intestine. But this also has a need to bring in glucose into the cell. For example, if all the glucose is being reabsorbed in this section, the cells further down would need to take glucose from the blood for their energy needs. Therefore, we have Glute 2, the bidirectional transporter, in the basolateral border of intestinal and renal epithelia. What about Glute 4? It is the insulin dependent transporter. And what do we mean by insulin dependence? The skeletal muscle cell, or the cells of cardiac muscle, or the brain or adipose tissue which have the Glute 4, do not constitutively express Glute 4 on the surface like it would happen for Glute 1. Glute 4 glucose transporters are located on vesicles in the cytoplasm, and only when insulin binds to its receptor on the cell, that insulin receptor complex would activate a set of signaling mechanisms in the cell which would cause the Glute 4 transporter to be translocated to the plasma membrane, and only then it is going to be available for binding glucose and taking it into the cell. Now, what is encouraging to know is that it is not only insulin that can do this. Exercise is another signal which can cause translocation of Glute 4 to the plasma membrane, even in the absence of insulin. In that sense, exercise is like insulin. It can substitute for insulin in removing blood glucose. And why is it important to remove blood glucose? High levels of blood glucose will cause glycosylation of proteins on the vessel wall, and that is bad. That will damage the blood vessels, and that is why you get microvascular disease in diabetes. Therefore, clearance of high levels of blood glucose by striated muscle through insulin dependent mechanisms is very essential for vascular health, and in that context we should understand that exercise can do this like insulin. We will conclude this session now. In this session, we have considered the kinetics of transport systems across the cell membrane and how we can differentiate simple diffusion via ion channels from facilitated diffusion via carrier-mediated transport processes. We also saw that carrier-dependent processes exhibit what is called saturation kinetics. From there, we moved on to see some details about facilitated diffusion. In forthcoming sessions, we will discuss secondary active transport and then move on to the ATP-dependent transport systems, which are primary active transport and ABC transporters. Thank you for choosing to watch this NPTEL lecture.